Hi, I'm Afify, and I will switch gears, so I will talk about something completely different. It's an ongoing project, so I will share with you some of the preliminary results and also how we are going to proceed from there. So as you know, mosquitoes transmit many diseases to humans, and this is done by blood feeding. They transmit malaria, for example, which is a big problem in sub-Saharan Africa. Dengue fever, which is now a problem in Saudi Arabia and in Egypt, but we don't have enough data to confirm this actually. Also, yellow fever transmitted by the same mosquito species that transmit dengue, West Nile virus, and filariasis, which is transmitted by Culex mosquitoes, which actually this is the house mosquito, so it's everywhere. Finally, Zika, which is all over the media now because it's responsible for microcephaly in newborns. So Zika now is all over the east coast of the United States, getting even to Baltimore. So we have been reported with some Zika cases in Baltimore. Malaria alone causes around 400,000 cases per year. So this made malaria is actually one of the most dangerous infectious diseases. The number of death cases that caused by mosquito-borne diseases in general are about 1 million per year. Unfortunately, some of these diseases have no successful treatment or vaccination. Even the ones that have, like malaria, it actually, there was some reports of drug resistance for the artemisinin-based drugs for malaria, for example. This makes controlling these diseases is easiest with using something to prevent bites, mosquito bites from the beginning, so you can actually stop the problem before it happens. So how we can do this? Mosquitoes find their host or the humans for blood feeding through olfaction or their sense of smell. So something around on our skin that attract them. But olfaction also can play against them by using repellents, for example, like DEET, Bicardin, or IR3535. These are common repellents that have been used successfully against mosquitoes. But unfortunately, these repellents have some health concerns. So DEET, for example, is prohibited in the European Union because of its effect on the nervous system. So what we want to do is to find alternatives to these repellents that work as efficient but also safe for human use. How we can do this? We want to understand first how these repellents affect the mosquito olfactory system, or in other words, how mosquitoes perceive these repellents. So first I will talk about how mosquitoes smell in general. So odor molecules in the environment are perceived on receptor cells in the antennae, maxillary bulbs, and proboscis of mosquitoes. I think I have a pointer here, I think. Oh, that's not a pointer. Okay. These receptor cells, they are housed in olfactory neurons in these appendages. Great, thank you. And these neurons send their axons to the brain. This is the first olfactory center called the antennal lobe, where they are processed and then send the information to higher brain areas for further processing. So think about it as a code. For each odorant in the environment, there are some receptors and also glomeruli in the brain that respond to that specific odor. And we want to understand the code in which mosquitoes understand odorants. So how we do this? We use something called calcium imaging. As you know, calcium is used for signal transaction, we want to measure how much calcium is inside the cell, so intracellular calcium, to indicate how the cell is active or not active. And we use what's called calcium indicators, like GCAM, for example. These are molecules that, when excited with a specific wavelength like this, it emits light at a different wavelength. But when they are bind to calcium, the light that is emitted is actually at a higher intensity, so they emit more light. And then we can perceive this light using just a fluorescent microscope, and then we can tell if a cell is active or not. In the past, and I did that during my PhD, we actually inject the brain of the insect with these molecules, and this is not efficient because each cell ends up taking a different amount of the molecules, and then we cannot compare actually between the cells. Now what we do is that we express these molecules in the olfactory system. So we make the cell produce these molecules themselves. So it's actually produced in the same amount in every cell. We use what is called the binary expression system, like the QF-QUS system, which is actually very famous. It's comprised of two components. The QF component, this is a transcription factor that is driven by a tissue-specific promoter. So actually this is expressed in specific tissue or our tissue of interest, which is the olfactory system. Then we use QS. This is the second component, the upstream activation sequence. When it binds to QF, it expresses the GCAM in all the olfactory cells. So what we actually did is that we produced a transgenic mosquito line that has the QF component in the olfactory system, and then another line that has the GCAM in the inactive state in all the cells of the body. When they are crossed genetically, we get a third line that has the GCAM in the olfactory system. So now we can use this line to test how repellents affect the olfactory system. And this is calcium imaging recordings. These are actual recordings. What we are going to see now is a video of the antennae of the mosquitoes in response to different odorants. So these are just short videos. So here, this is a response to mineral oil. When the red rectangle comes, there should be an odorant that is delivered, but you cannot see anything because mineral oil is actually not perceived by mosquitoes, so they cannot smell it. So this is our control. But if we used a repellent like mineral grass oil, we see that there are some cells that is activated. These are the cells that is responsible for coding mineral grass oil. We can quantify this, and we can tell that there are 3 cells that respond to the odorant. And here, this is another odorant, eugenol, that respond to, that activate 2 cells for a long time. So here comes the odorant, and it's actually 1 second of the odorant activate the cells for 50 seconds. So this is actually abnormal activation. Then DEET, for example, and DEET is one of the most common or the commonly used repellent. It does not activate the cells, so we don't really know how it actually affects the mosquito olfactory system. So we tested this compound with another compound in a mixture, and we found that it masked the odorant. So actually what it does, it masks our smell, so the mosquito cannot see us or they cannot detect us. So this is the same with another attractant, cyclohexanone. Now what we do is that we categorize the repellents into 3 categories. Like lemongrass oil, for example, activates a subset of receptor neurons. There are repellents that activate for a long time, like eugenol, and there are repellents that just mask or decrease the response to attractive odorants. Next, we want to find out how or which receptors activated by different repellents. So we extracted the cells and we sent them for microarray analysis, but we couldn't see any signal because we actually extracted a number of cells that is neighboring to these cells, so we get a lot of noise. But what we do next is we will use what's called the laser capture microdissection. So it's a microscope equipped with a laser beam, so the laser comes from beneath the sample, and then it cuts around the cell, and we can extract that cell, and we send it for microarray, so we can tell which cell is responsible for the repellent. Then we will use the famous CRISPR-Cas9 technique to knock out the receptor, and then we have a mosquito line that lacks that specific receptor. This way we can test these mosquitoes behaviorally, like in this chamber, for example, or what's called a landing assay, and we can tell if the repellent is actually perceived with this specific receptor or not. In the end, or this is the end goal, is actually to screen for many odorants using this receptor and find out the ones that behave the same way as the strongest repellents. And these will be just the efficient repellents that we can use on a daily basis with different mosquito species. So at the end, I would like to thank my group members at the School of Medicine at the Department of Neuroscience, the NIH, the Bloomberg School of Public Health, Johns Hopkins, and the JHMRI for funding my research, and of course Fulbright for giving me the opportunity to talk to you today, and you, of course, for listening. Thank you.